Speaker. Dr. Robert M. Califf is the Commissioner of Food and Drugs, and as the top official of the FDA, Dr. Califf is committed to strengthening programs and policies that enable the agency to carry out its mission to protect and promote the public health. Dr. Califf also served as the FDA's Deputy Commissioner for Medical Products and Tobacco from February 2015 until his first appointment as Commissioner in February 2016. Please let us welcome Dr. Califf.
Thank you, Brenda. I'm pleased to join everyone for this important workshop designed to stimulate and support innovation in generic drug development. My appreciation to all the developers and other stakeholders who've joined today's discussion. I also want to thank industry for its support of the Generic Drug Development Program, without which this meeting would not be possible. This is the 5th year we've held this workshop. It's designed to provide you with a variety of opportunities to hear from FDA staff on recent innovations in science and cutting-edge methodologies behind generic drug development, issues related to product-specific guidance development, as well as common issues seen in abbreviated new drug applications, or ANDAs, and pre-ANDA meeting discussions, all to help you produce more complete and responsive drug applications and more first-cycle approvals. During the workshop, you'll be hearing from FDA experts on a range of topics, including peptide and oligonucleotide active pharmaceutical ingredients, or APIs, sameness, and impurities, drug-device combination products, long-acting injectables, oral complex drug products, nasally administered products, quantitative methods, and model-integrated bioequivalence approaches. You'll also get some insight into upcoming GDUFA III enhancements. In short, this program is designed to help you, as the title underscores, to translate science to approval. Indeed, that process is one of the most essential, as well as one of the least understood of the FDA's roles, to help make sure that promising scientific developments get translated into meaningful products that can make a difference for and are readily available to the patients we care about. The primary way we do this is through regular communications with developers. FDA staff have a wealth of knowledge to support the work of developers. Through these communications, especially during the pre-market phase, we can provide greater regulatory clarity and help direct, support, and advance development programs. We bring the awareness and understanding of the success and failure of all products in development. This can help guide developers to the best and most efficient pathway, saving time and money, preventing wasted or duplicative efforts, ensuring speedy development of the best and most effective products, and reduce risk to human research participants. The FDA's involvement with developers is particularly important in the generic drug space because of the enormous benefits these products provide to patients and consumers. As we know, competition from generic drugs helps lower drug prices. According to the Association for Accessible Medicines, in 2020, the healthcare system saved $330 billion thanks to generic prescription medicines. And Americans clearly are taking advantage of these savings. Generics today represent 90% of prescriptions filled, accounting for 18% of all prescription drug spending. As I turn 71 this month, I'm glad to have access to generic medicines for my blood pressure and lipids. It's a fair deal to have low-cost access after a period of exclusivity. What these numbers mean is that more patients have greater access to affordable, safe, effective, and high-quality medicines, and that's a priority for the FDA. Indeed, we won't let anything sidetrack us from meeting this goal. Consider our work during the COVID-19 pandemic. Since the pandemic began, CDER has approved more than 1,700 COVID-19 related ANDA approvals, including critical medications that have been in shortage due to their use in treating patients with COVID-19. The Office of Generic Drugs partnered with the Office of Translational Sciences and reviewers and project managers in the Office of Pharmaceutical Quality to discuss and complete these ANDA reviews. And even with this increased focus on COVID-19 related ANDAs, the generic drug program kept up with the normal flow of hundreds of original approvals and thousands of supplements, meeting nearly all of our generic drug user fee amendments performance goals. So how do we build on this? We know that future savings and the viability of the generic drug program depend on industry continuing to invest in developing products. And the FDA must support this by building and maintaining an efficient and transparent system for the generic drug program. During today's workshop, you'll hear about a number of ways our generic drug program continues to modernize generic drug product development and enhance our science, policy, and business processes. For example, we're modernizing our science to develop efficient bioequivalence approaches for products that eliminate unnecessary human studies and provide approaches for generics for complex products. We're modernizing our policies to help industry maximize the use of the 505J pathway and have greater clarity around how to develop generic versions of drug-device combination products. And we're modernizing our processes to increase pre-submission interactions with industry to make each submission more complete and our reviews more efficient. There is, of course, another key piece to building a modern future, which is GDUFA. The GDUFA Science and Research Program enables the FDA to address complex scientific issues for product-specific guidance, to develop and communicate with applicants through the pre-ANDA meetings to help clarify regulatory expectations for prospective applicants early in the generic product development cycle. These early communications can help reduce a generic drug product's time in the pipeline from concept to development to market by assisting generic drug applicants in developing more complete application submissions. Since 2012, GDUFA has played an important role in supporting the development of generic drugs, including complex generic drugs, and making their review more efficient by proactively addressing emerging scientific and regulatory issues. As you understand, complex products are critical to the treatment of many medical conditions, but because they can be more scientifically challenging, time-consuming, and expensive to develop, these drugs can lack adequate generic competition. Consequently, they, along with other products, where there is uncertainty concerning the approval pathway or possible alternative approaches, can benefit from early scientific engagement. Let me give you a quick and noteworthy example of how this can work. This past February, the FDA approved the first generic of Restasis, cyclosporin ophthalmic emulsion, to help treat a type of dry eye syndrome. That approval followed more than 10 years of GDUFA research, which successfully addressed challenging product characterization issues, developed analytical measurement and statistical analysis tools, and supported updates to generic product development recommendations in product-specific guidances prior to the assessment and approval of the first generic product. And today, thanks to that research, we have an important and affordable generic treatment that's readily available. With the expiration of GDUFA II, we're working on a number of enhancements for GDUFA III intended to further enhance efficiency, transparency, and gain more first-cycle approvals. These include additional scientific meetings with industry, both prior to and post-submission, so that applicants can communicate with the FDA in a timely manner for input on their development program to improve efficiency and enable better ANDA submission. Let me mention one other point before I let you get to the rest of the meeting. As you all well understand, drug development today is global in nature. Consequently, generic drugs are quite often the product of a global supply chain and produced with the intent to market in multiple jurisdictions. Last year, the FDA launched a generic drug cluster, which offers a forum for the world's leading regulatory agencies to better enable harmonization by early communications and collaborations related to generic drug development, especially related to individual drug products and approval. And it will also help ensure that the benefits Americans get from generic products are not easily disrupted in the event that some global event, whether due to natural disaster or international strife, upsets the supply chain. Before finishing, I want to emphasize 2 points that I believe should be a priority for all of us. First, if you're comfortable, please get your booster and encourage your friends and family to do so. In fact, if you want to see all your family during this year's holiday season, getting the latest updated booster is probably the most important single thing you can do. The outcome data demonstrating the life-saving impact of these boosters is overwhelming. Second, please engage with others on the issue of truthful and reliable scientific and medical information. We're currently seeing too many people get sick or die because of bad decisions prompted by naive or purposeful misinformation. We all need to work together to improve the level of trust in science and in medicine, and we all need to be active in promoting truth. I want to thank all of you again for your participation today. This workshop is a critical part of the pathway for industry in supporting innovation to lead to the development of new products. When that pathway is unimpeded, the FDA and industry are able to join together to deliver timely, high-quality, safe, effective, and affordable generic products to the American public. That's our goal, and I look forward to continuing to work with you to achieve it. I hope you have an interesting and productive meeting.
Thank you for the great keynote presentation, Dr. Califf. I'm Ray Ford, working with SBIA, and I'll serve as your moderator. It's my pleasure to first introduce our session leads. First, we welcome Dr. Darby Kozak, the Deputy Division Director for the Division of Therapeutic Performance 1 and FDA's Office of Generic Drugs. Dr. Kozak leads a group of interdisciplinary scientists on the development of new analytical methods and equivalence evaluation methodologies for complex drug substances and parenteral, ophthalmic, and otic formulations. Our second session lead we welcome is Dr. Cameron Smith, who's a branch chief in the Office of Life Cycle Drug Products, Office of Pharmaceutical Quality in Cedar. Our first presentation is on the guidance for peptide products and assessing immunogenicity risk. Our presenter, Dr. Eric Pang, serves as a senior chemist in the Office of Generic Drugs. He specializes in the analysis of peptide and large molecule drugs. Currently, he serves as the SME for peptide drug products, develops of product-specific guidance of complex drug products, responds to controlled correspondences, pre-ANDA meeting requests, citizens petitions, and consultations. Our second presentation is on the common deficiencies associated with comparative peptide impurity profile studies and qualification of impurity levels and proposed limits. Our presenter, Dr. Yili Li, joined FDA in 2016 as a chemistry reviewer. She has served as a primary reviewer, secondary reviewer, and application technical lead in the Division of Liquid-Based Products 1 for ANDA and pre-ANDA applications. She has extensive experience in the review of complex peptide drug products, topical products, and parenteral products. Our final presentation in this session is on the assessing impurities to inform peptide immunogenicity risk, developing informative studies. Our presenter, Dr. Daniela Berthelli, currently heads the Laboratory of Innate Immunity and chairs CEDAR's newly formed Center for Excellence in Infectious Diseases and Inflammation. Please join me in welcoming our first presenter, Dr. Eric Pang.
Thanks for the introduction, and thanks to the organizer for giving me the opportunity to share this presentation with you on assessing immunogenicity risk of peptides as recommended in the synthetic peptide guidance and PSGs. First, I'll introduce the peptide guidance for certain highly purified synthetic peptides that reference the listed drug of our DNA origin, where the concept of immunogenicity risk assessment for peptide products are introduced. I'll clear up some common misunderstandings of this guidance. Then I'll give an overview of immunogenicity and non-clinical assays. Next, I will discuss a little bit on the PSG development for other peptide products, specifically those not outlined in the aforementioned peptide guidance. Finally, I will mention briefly on the immunogenicity risk assessment for peptide products, which is a topic you'll hear more in Daniela's talk later as well. As many of you know, peptides can generally be manufactured through the following pathways. A chemical synthesis, which is a step-by-step addition of amino acids using chemical reactions, and recombinant expression, which is recombinantly express the peptides and then extracted from yeast and bacteria. Maybe there are a few that can be extracted from natural sources as well. The different manufacturing process can result in different impurities, which may give rise to different safety concerns. These impurities can generally be characterized into either process-related impurities, such as wholesale proteins, leachable, extractable, microbial contaminants, and etc. Generally speaking, the chemical synthesis overall has a lower risk because of the lack of these process-related impurities, such as wholesale proteins and microbial contaminants. But it does still have other impurities that may be present. Then there's also peptide-related impurities, and these can be generally referred to as impurities related to the API peptides, such as impurities that carry a deletion, a duplication, or modification of the sequence. Therefore, a proposed generic should demonstrate that the difference in impurities will not increase its product risk. And it is with these considerations in mind that we published the guidance on ANDAs for certain highly purified synthetic peptide drug products that refer to the listed drugs of rDNA origin, and later I'll refer to as the synthetic peptide guidance. This guidance outlined the agency's current thinking on addressing potential immunogenicity risk for synthetic glucagon, liraglutide, nesiratide, teriparatide, and tetraglutide, these 5 peptides referencing recombinant RLDs. In order to ensure that the proposed synthetic peptide product is sufficiently similar to the RLD when it comes to safety risk, it is recommended that the following with regards to specified peptide-related impurities. The specified impurities that are commonly found in both generic and RLD, the level of these in the generic product should be no more than the RLDs. And for new impurities in the proposed generics, like those that are not found in the RLDs, it is recommended that these impurities to be not more than 0.5%. In addition, these new impurities that are found between 0.1 to 0.5% should be identified, characterized, and justified for not affecting the safety and efficacy of the proposed product, which we recommend to do by conducting comparative immunogenicity risk assessment. Next, I'll make a few clarifications when it comes to applying the previously mentioned synthetic peptide guidance. First, please understand that like all product-specific guidance, the ANDA synthetic guidance contains non-binding recommendations. Another common misunderstanding is that the synthetic ANDA guidance applies to all peptide products, and that is not true. The synthetic ANDA guidance should only apply to the 5 peptide products that are outlined in the guidance. However, depending on the risk of the peptide product, the immunogenicity risk specifically, some of these principles and recommendations outlined in that guidance may be borrowed. So I'll discuss this point a bit more in detail when we come to the product-specific guidance. And finally, the question about whether ANDAs having new impurities greater than 0.5%. And for these impurities, we think it may be issue if we rely on the non-clinical assays for risk assessment. So typically, we will recommend that the applicant to work on the manufacturing and control to bring down the impurities that are greater than 0.5% to less than 0.5%. However, for some reasons, if these impurities cannot be resolved, that they cannot be brought down to a sufficient enough level, we ask the applicant to bring this to our attention and reach out and discuss this issue with us through control correspondence or other meeting requests. Next, I'll discuss impurity-related immunogenicity risks. I'll start the discussion with a general overview of the human immune system. Immunity is a complex system with many interplaying components. So to realize that this is a rather simplistic view of the human immune system here. In general, the immune system can be divided into two main aspects, innate and adaptive immunity. To the left are the cells involved in the innate immune response, which tend to be the first responders to the presence of a foreign invader or an infection, and in the drug product, these process-related impurities such as contaminants and leachables that may trigger innate immune response, hence the name innate immune response modulating impurities, or short for IIRMI. To detect the immunogenicity potential or the presence of these product-related impurities that triggers innate immune or inflammatory response, in vitro assays are available and can be used to detect these on the product as a whole. And to the right are cells involving adaptive immunity, which also known as acquired immunity. In adaptive immune response, the antibodies are created to target specific pathogens, and this is done by recognizing specific sequence or regions of the antigen. So in peptides, the applicants are asked to investigate if for example, new impurities, whether these new peptide-related impurities, such as impurity containing a deletion or duplication of the amino acids, do contain the MHC binding domain. And to understand if any of these peptides-related impurities can contain the MHC binding domain, in silico and in vitro assays can also be used. So next I'll give a general overview of these assays. An example of these adaptive assays for assessing adaptive immune response are the following. There's the in silico immunogenicity risk assessment of the MHC binding. The advantage of using in silico is that it is a quick way to screen and predict the presence of the binding epitopes without the need to individually isolate impurities and experimentally test them. However, one realized that the in silico immunogenicity assessment should be complemented with the in vitro study as well. The in vitro assays, there are a number of those, and here I outline two of them, which is HLA binding study or cell-based assays such as T-cell proliferation assays can also be used. And then there's if one chooses, you can also choose animal models such as the transgenic mouse model. And for the in vitro assays for assessing innate immune response, here's a table. And we have a number of these cell lines, such as the PBMC, the raw blue, macrophages, and so forth. These are, the good thing about these cell lines are they are commercially available. The PBMC and whole blood will need donations from human blood, but all of these are commercially available, and they do test different components or responses of the innate immune response. And Daniela, I know later will discuss more in depth about these assays. So I will refer you to her talk in the later of the session. These non-clinical assays are relatively new to many generic applicants. So it's not surprising that we see some of the common challenges shared by many of the applicants when these assays are reviewed. Some of these common challenges include typically we see insufficient demonstration of the assay sensitivity. there's insufficient justification of the type of assays or the methodology used. And also, just in general, not enough detail has been provided on the method. For example, the concentrations tested, number of subjects, and etc. So again, Daniela will provide some of the details about running these assays in later of the session. So I refer you to her talk. So besides the synthetic peptide guidance, which covers the 5 specific synthetic peptides, we also publish and still publishing PSGs for other peptide products. Similar to the other PSGs that we publish, we develop these PSGs with the current thinking and information known about the studies that are that's needed and recommended to support the generic approval. So for example, there are studies that demonstrate the pharmaceutical equivalence or studies demonstrate bioequivalence. And per GDUFA II commitment letter, the agency agreed to publish PSGs for new complex products as soon as scientific recommendations are become available. And the PSGs recommendation for peptide products is actually based on the consideration for immunogenicity potential as well as demonstrating the product's sameness. So in this table, I'm showing a few examples of the peptide PSGs that we have published. And looking at the recommended studies in these published PSGs, we know that, as you can see, all of these studies, API sameness, impurity profile, analysis, adaptive immune, innate immune, high order structure, ligaments, and biological activities. All these are studies that are recommended in the synthetic peptide guidance. However, not all peptides would have recommended all these studies to, as you can see, it really depends on the risk of the peptide. For example, the semaglutide. Semaglutide is rather quite similar to liraglutide, so one would think, based on its similarity with the indication, also the structure of a liraglutide, it probably carry about similar kind of risk. Therefore, it will recommend pretty much all the studies that are recommended in the general synthetic peptide guidance. So that's why in this particular PSG for semaglutide, we simply reference the peptide guidance. But for vasopressin and secretin IV solutions, these believe have less immunogenicity concern in comparison to the ones that are outlined in the peptide guidance. So for these, we treat them as we'll recommend the sponsor to do immunogenicity impurity profile first. And if we do see any impurities that are unusual, then we will recommend immunogenicity study. And for the bremelanotide and octreotide sub-Q solutions, these have even less of an immunogenicity concern because of their small size and also various other reasons. So for these, we just recommend that the peptide should have demonstrate the API sameness as well as a high order structure analysis. So as you can see that just because it's a peptide, we're not going to recommend all of these studies that are in the synthetic peptide guidance. So it really depends the type of peptide and the potential concern and risk that this peptide may have. And we'll talk about, and I'll talk about a little bit about the risk assessment, immunogenicity risk assessment of the peptide next. So when we talk about the immunogenicity risk assessment, this is what we're looking at when we assess the risk of a peptide product. It is based on the ROD, the peptide product, its size, its route of administration, dose frequency, how similar it is to human the endogenous human proteins, its half-life in the system, and so forth. And then there's the intended patient considerations such as its indication, the immune status of the intended patient, and so forth, and then our clinical experience with the ROD, such as the observed anti-drug antibody level found during clinical studies, any adverse event that's related to immunogenicity, and so forth. So this, I believe, will also be discussed further later in the session. So in summary, the PSGs contain recommended studies for these peptide products, and the type of studies recommended depend on the current thinking and understanding of the associated risk of the peptide products. The synthetic peptide guidance targets specific 5 peptides where the immunogenicity is a concern. And currently we are developing PSGs for other generic peptide products as well, and we publish some of those. Non-clinical immunogenicity assays can be used to assess the comparable risk of the generics compared to the RLD. And later on in the session, you're going to hear more about these non-clinical immunogenicity assays. So finally, we recognize that these assays are relatively new to the field, and so there's a need to establish the best practice and standards for conducting these assays. So it is with this in mind that we are conducting additional GDUFA funded research projects. And because there's a need, that we recognize there's a need for developing the assays for non-clinical assessment of the immunogenicity, we have the following GDUFA funded research looking into this. Recently, in 2021, we published a paper looking at the innate immune response assay using PBMC and how to best handle these cells, and published in the Journal of Molecules. And then we have other contracts that are ongoing as well, looking at the in silico in vitro assessment of the adaptive immune response. We are still looking for additional collaborators and researchers to help us better evaluate and develop these assays as well. So we encourage you to, if there's an interest to looking into these assays, we encourage you to submit research proposals through the GDUFA funded research portal here at this address. And with that, I'd like to end with acknowledgement our office, ORS, and also our collaborators at OBP and OLDP. Thanks for your attention. So now we come to the challenge question. Challenge question 1. Which of the following peptide product is not covered by the guidance, and that's for certain highly purified synthetic peptide drug products that refer to listed drugs of rDNA origin? A. Neseritide, B. Tetraglutide, C. Glucagon, and D. Secretin. And if you pick D, that is correct. Secretin was not one of the 5 peptides. And challenge question number 2. Which of the following statement is not true? A. PSGs contain recommended studies to demonstrate product sameness for both pharmaceutical equivalents and bioequivalents. B. Immunogenicity risk assessment using non-clinical assay is recommended for all peptide products regardless of their risk. And C. Adaptive and innate immune response assays are typically recommended for certain peptide products with immunogenicity risk concern. Or D. Peptide products not covered by the synthetic peptide guidance may still reference part of the recommendations outlined in that guidance. And the correct answer is B. Thank you for your attention. And I'll take your questions at the discussion panel.
Good morning, everyone. My presentation today is common deficiencies associated with comparative peptide impurity profile studies and qualification of impurity levels and proposed limits. My talk will cover learning objectives, introduction, quality considerations associated with peptide-related impurities, aggregates, immunogenicity risks. My talk will only focus on those peptides covered by the peptide guidance and those that immunogenicity might be a concern. I will then present some common deficiencies and a summary. In my talk, I will describe regulatory challenges for synthetic peptide generic drugs, such as how to mitigate risks for peptide-related impurities and risks for higher molecular weight peptide aggregates. I will then review examples of common deficiencies to highlight aspects where attention should be focused to facilitate ANDA approval. For small organic compound drugs, we usually use the product identification and qualification thresholds to set up impurity limits. However, peptide drugs are specifically excluded from ICH Q3A Q3B guidelines. So how to qualify impurity levels and set up appropriate limits become a regulatory challenge. Additionally, peptide-related impurities and aggregates may pose immunogenicity risks. Given this complexity, it is important to focus on those attributes to facilitate peptide ANDA approval. According to the ANDA peptide guidance, for a peptide ANDA application, it is required that you establish the API sameness of the generic product and the reference product. It is also required that you perform peptide-related impurity profile comparison studies. API sameness can be established by characterization of primary sequence and physicochemical properties, oligomer aggregation states, secondary structures, and biological activities. Today, I will only focus on oligomer aggregation states and the peptide-related impurity profile. Qualification of impurity and aggregation levels should be based on the comparative characterization results of the test product and the reference product. For comparative study of the impurity profile, it is recommended that you use orthogonal chromatographic methods with different separation principles, and also use high-resolution MS and MS-MS for peak identity and matching with ROD. For comparative study of oligomers and aggregates, again, orthogonal methods should be used. Study should be directly on the drug product formulation. If it's not feasible, it is recommended that you use minimal sample manipulation, and also provide a justification as why you choose the proposed study condition. We also recommend that you perform the study under stress conditions, especially when aggregates are not so significant under normal storage condition. You need to demonstrate that under stressed conditions, the test product is still comparable to the ROD. It will also help to demonstrate that your analytical methods are capable of detecting aggregates if they are present. During the impurity profile characterization, you need to identify each peptide-related impurity at the level of 0.10% or higher. Demonstrate no peptide-related impurities are higher than 0.5%. For impurity found in both the test and ROD products, demonstrate the level in the test product is the same as or lower than that of the ROD. Otherwise, if you observe an impurity between 0.10% and 0.5%, and it is either not present in the ROD or in the test product at a higher level than that in the ROD, then you need to provide justification as why such impurities does not affect the safety, effectiveness, or potential for immunogenicity. For impurity limits, we expect that you set up the limits based on your actual stability data. Therefore, qualifying impurity level and setup specification limits should be the same thing. The limits should be set up at either less than 0.10% or qualified by observed ILD levels, or the proposed limit is between 0.10% and 0.5% and supported by immunogenicity study results. If you set the limit higher than 0.5% and not supported by the observed ILD level, probably due to your stability data, please refer to the talk by Eric Pan. Basically, we encourage you to contact the agency up front to make sure your filing pathway is appropriate. In general, we are talking about the generic formulations that are Q1, Q2 similar to the ILD. So it is expected that the test product has similar aggregation profile as the ILD, which means that the aggregation level in the test product should be qualified by the observed ILD levels. To mitigate the risk of the immunogenicity caused by aggregates, you need to perform innate immune response studies. These studies should, in the meantime, mitigate risks for other product and process-related impurities. I will talk about this again in the following slide. In the cases that a generic product contains a greater level of aggregates, then an investigation should be performed to identify the root cause and implement appropriate manufacturing process control as necessary. To mitigate the immunogenicity risks of the test product, first of all, you need to perform innate immune response evaluation on the fully formulated drug products. The purpose of this study is to ensure the generic product does not contain anything such as impurities, aggregates, contaminants, or leachables that may increase the immunogenicity risks. Then, if the tested product contains any new or elevated impurities that are between 0.10% and 0.5%, adaptive immunogenicity risks for such impurities should be evaluated individually. In other words, the study should be conducted on the purified individual impurities. Sample requirement. For comparative studies of impurity profile, aggregation profile, and innate immune responses, please make sure that the study are conducted on at least 3 batches of the test product that are manufactured from at least 2 batches of the drug substance, tested on or near release and at the end of shelf life. Also conduct the study on at least 3 batches of ILD of different ages prior to expiry as available. Please provide a table indicates the drug substance, drug product batch number, manufacturing date, and testing date to make it clear the sample ages used in your studies. Now I'm going to use some common deficiencies as examples to explain certain points in more detail. In your original submission, you probably only have impurity characterization data on the test samples aged near release. You will be asked to update the study with samples aged at the end of your proposed shelf life. If you only developed related substance method to perform the study, you will be asked to perform the study using orthogonal methods that use different chromatographic separation principles. During the impurity profiling comparison, you need to provide the peak identity information for the test product and the RD because only retention time is not sufficient to conclude that the test product and RD have the same impurity. Also, we need to know whether each peak contains single impurity or multiple co-eluting impurities. So for this, high-resolution MS or tandem MS are recommended. We quite often see applicants proposing to control some of the specified impurities as mixtures in the drug product specification due to co-eluting. It is recommended that you improve the analytical method to resolve all specified impurities and control each with individual limits, or use multiple methods to control subsets of impurities. Controlling impurities individually will help you to qualify your proposed limits. For example, if impurity 1 is qualified to a limit of 0.1% and impurity 2 is 0.5%, when you control them as a mixture, the limit for the mixture can only be 0.1%. So you may end up having difficulties in controlling impurity 2. We still see applicants trying to identify some impurities in the specification table with relative retention times. This is unacceptable because per the peptide guidance, you are required to characterize everything at or about 0.10%. So you should provide a name and a structure for each impurity in the specification table. When you propose a limit of no more than 0.10% for any unspecified impurity, we usually recommend you to tighten the limit to less than 0.10% to be consistent with the guidance. If your proposed limit is higher than 0.5% for certain impurity and the limit is not supported by the observed RD level, you should tighten the limit to be consistent with the observed RD level, or tighten the limit to between 0.10% and 0.5% and supported by immunogenicity study results. If above are not achievable due to your stability data, you may need to discuss with the agency before you end up filing. If you reported a greater level of aggregates in the test product than those found in the RD, you should perform an investigation to identify the root cause and improve your product aggregation profile through implementation of manufacturing control as necessary. You should also demonstrate your improved manufacturing process is capable of producing drug substance drug product batches with acceptable aggregates control across the proposed shelf life. Now comes to two challenge questions. I hope you all will get it right. Challenge question 1. Consistent with the end-peptide guidance, when conducting a comparative impurity profiling of the generic product and ROD, identify all peptide-related impurities at a level of A, higher than 0.10%, B, higher than 0.5%, C, at or higher than 0.10%, D, at or higher than 0.1%. 4 seconds. The correct answer is C. You need to identify all peptide-related impurities at a level of 0.10% or higher. Challenge question 2. Which of the following statements is not true? A. ICH Q3A Q3B guideline is generally applicable to all drug products, including peptide drug products. B. The limit for each peptide-related impurity can be qualified by the observed IOD level for the same impurity. C. A comparative characterization of the impurity profile of the generic product and IOD is required to establish acceptance limit, impurity limits. C. Peptide-related impurities and aggregates may pose immunogenicity risks. Again, 4 seconds. The correct answer is A. ICH Q3A Q3B guideline is not applicable to peptide drug products. In summary, I provided an overview of some of the regulatory requirements for synthetic peptide generic drugs covered by the peptide guidance. The requirement of comparative peptide-related impurity and aggregation profile characterization studies. I also explained how to qualify impurity levels and propose specification limits based on the comparative impurity profile data. I finally give some examples of deficiency language commonly communicated to the ANDA applicants regarding compliance with the ANDA peptide guidance. I want to thank my branch chief, Cameron Smith, my division director, Binkan, and my previous branch chief, Pahala Simora, for their valuable inputs and comments. I'll be happy to answer your questions during the panel discussion. Thank you for your attention.
Thank you. My name is Daniela Bertelli. I'm from the Office of Biotechnology Products, and I would like to focus now your attention on some of the key aspects of the assays that are being used to inform the immunogenicity risk of generic peptides. As Eric mentioned in the first talk, there are a number of non-clinical studies that can inform the immunogenicity risk assessment for some generic peptides. Now, to be very clear, for the first purpose of this talk, it will be a given, not only that the product will be used by the same population in the same manner as the RLD, but also that the sameness of the API has already been established, right? And so if the sameness of the API has already been established, we can then really think about the impurities as the major factors of any residual uncertainty, and we can use non-clinical studies to assess those. Now, why do we need to characterize them? Because process-related impurities can activate innate immune cells in the tissue where the product is being delivered, and peptide-related impurities can offer new epitopes to be presented by MHC-bearing T cells. And these two factors are really going to impact on the immunogenicity risk. The thought process behind this is outlined, as Eric mentioned, in the FDA guidance on synthetic peptides that focuses on the risk of glucagon, liraglutide, nesirotide, teriparatide, and teduglutide. And once again, it is based on the products having the same API and the understanding of the prior clinical experience and the risk assessment of any potential impurity. Why do we focus on product and process-related impurities? As previously explained, tissues such as the subcutaneous space contain many antigen-presenting cells that actively sample their environment and can be activated by innate immune response modulating impurities that are co-delivered with the product. Their activation leads to an influx of inflammatory cells to the site, and the relocation of these antigen-presenting cells to the draining lymph node, where they can present the peptide, whether it's the API or peptide-related impurities, to naive T cells, initiating an immune response. Further, the type of activation of the antigen-presenting cells will determine the characteristics of the immune response elicited. So when we want to assess what are the process-related impurities, or whether there's any difference in process-related impurities, we really, our focus is on whether there are any innate immune response-related impurities. Those studies are done in vitro using the whole drug product, although they can be supplemented with some studies on drug substance law. When we're talking about peptide-related impurities, then the focus is usually on conducting in vitro studies on purified impurities and understanding what is their capacity to bind MHC, is their potential tolerance, what is their T cell stimulating ability. Now, let's start with the assays that look for innate immune response modulating impurities. These are assays that are usually focused using either PBMCs, whole blood, or reported cell lines that are put in cultured in the presence of drug product. They're incubated, and the readout is either NF-kappa B activation, cytokine production, cell microexpression, or messenger RNA. It is very important that these assays have useful suitability controls. The intent of these assays is to detect biological differences in immune markers, with the idea that an increase or a shift in the biological response could suggest an increase in clinical differences. Now, very importantly for synthetic peptides, the expectation is that there are no innate immune response modulating impurities. And to demonstrate the absence of those impurities, then it's very important to have well-qualified assays and that those assays are highly sensitive. Now, these assays, as I was saying before, they can be done with whole blood, with PBMCs, dendritic cells, or they can be done with cell lines, whether it's cell lines expressing a variety of receptors or cell lines that express, very targeted receptors, single receptors. And there's a variety of those in the market. The readouts, as I said again, can vary. They can be proliferation cytokines, activation markers, NF-kappa beta activation, and each of these systems have advantages and disadvantages. When we're thinking about primary cells, it is very important to understand what are the critical assay attributes. So if we're thinking about primary cells, please tell us, what number of PBMCs or what number of donors was used, and what are the qualifications of those donors? Are they healthy subjects? Are they disease subjects? How have the samples been processed? How have the samples been selected? What is the cell viability pre and post assay? Have you made certain that you have a certain percentage of viable APCs in the mix so that we understand what we're looking at? And here on the right is really an example where you can see that the response in terms of IL-1 and prostaglandin E2, showing the response of fresh PBMCs versus whole blood culture versus PBMCs that have been stored for 24 or 48 hours. And you can see that the culture conditions makes a huge impact on whether those differences can be detected and the magnitude of those differences. When we're thinking about cell lines, again, what is the coverage? What is the receptor coverage in terms of innate immune receptors? You have to be very aware that cell lines usually have a very limited sensitivity to product aggregates, and their acceptability is really going to depend on product risk, right? What is the risk associated with the product might determine whether cell line data alone is sufficient to understand the risk. And then we're going to need to understand other factors. What are the culture conditions? How many cells per well? What's the culture time? What's the media? Is there any matrix interference? And we just published a manuscript showing issues formulation and excipients, how that can impact on this type of assay, and what is the drug concentration as well, also very important to know. So other important characteristics that need to be shown so that we can understand that the assay is suitable are, of course, what's the sensitivity, drug tolerance, specificity, precision, robustness, accuracy. Think ICHQR, right? Think what are the cell number, the viability, the composition of the cells that are in the well, what's the cell passage, if it's a cell line, how is the drug formulated? All these are going to be information that we need to have. What suitability controls are being used for negative controls, low and high positive controls, and can you demonstrate that consistent sensitivity to low levels of a variety of innate immune response modulators that are capable of triggering diverse innate immune pathways? Can you demonstrate really signal recovery? If you spike the drug prior to any dilution, prior to any manipulation of the drug, can you still recover? Is your assay sensitivity really what you think it is? And then in terms of the output of the assay, we usually prefer multiparametric assessments than looking for a single parameter. There's a number of different parameters that have been used in the field. There's NF-kappa beta, there's DC activation, cytokine expression, gene expression patterns. In our experience, when you have multiple parameters, you can begin to see different, and you look at them together, sort of you can begin to see differences between products that might not be evident when using a single readout. So usually a comprehensive parameter assessment is preferred since impurities can trigger different innate immune paths capable of increasing the immunogenicity risk. Now, many sponsors sometimes wonder, what level of sensitivity is sufficient? And that's a great question. There's really little data on this. But what we've been using so far as a guide is the levels of TLR agonists that have been shown to stimulate an innate immune response in primates, sort of as a minimal level of sensitivity that should be achieved. I'm not going to go over this table, you have it in your materials, but please bear in mind that not all sources of purified PRR ligands are created equals. So you need to justify your selection and qualify the materials that you are using. And of course, you need to time and again demonstrate that the sensitivity of the assay is reproducible, and you can recover the signal when you spiked the product into the impurity, the model impurities into your product. Now, so in terms of assay characterization and the results and the data that we need to receive, so it includes the SOP, cell isolation method or cell passage number, final concentration of drug product in the well, and any drug product manipulations that you do. Validation studies demonstrating sensitivity, LOD, LOQ, linearity, precision, etc. And basically what we need is data demonstrating that the assay is fit for purpose, and then provide the result. With the results, provide results of at least 3 batches of drug product at release and at end of shelf life. So again, preferably from different drug substance, from different manufacturing campaigns. We want to have as comprehensive an idea of the potential impurities as possible that you may have in your product. Again, relevant cell recovery and viability in the assays you conducted, data from suitability controls from each run, confirming the responsiveness of each donor or each cell line run, data from each run confirming assay sensitivity, numerical results, and not positive and negative. And I can't stress this enough, and there's been some confusion thinking of maybe anti-drug antibody assays where you sort of have a cut point and discern subject as positive, negatives. That's not what we're interested in here. We're interested really in the magnitude and the characteristics of the response. So identifying samples as positive or negatives doesn't really tell us enough. Common deficiencies, things that we've seen so far. Inadequate assays, either because sensitivity of breadth of receptors that are covered, the assays sometimes are not adequate. Problems with the characterization of the assay that include either number of donors, the donor selection criteria, cell numbers, duration, culture conditions, use of the assay, inappropriate stability controls, inadequate number selection or information on the drug product lots, excessive drug product dilution. This is something that we see very commonly if you're looking for something that's present most likely in a very small amount. Then if you really dilute your product, you're not going to really detect the impurity. And I try to really illustrate this with this triangle. So you know, there's going to be a dilution where you really can't use to detect impurities because the product at a very high concentration might interfere with your cells. And there's going to be an area where we really can't detect impurities because the product is too dilute. So you really need to look for that green sweet spot where the assay really works. Now let's move to the peptide-related impurities. And I really want to stress how important this is. And I thought that this was really a good example. You can see the difference between taspoglutide and semaglutide. And it's really a very few amino acids, and a side chain is the whole difference in terms of structure, but it makes a huge difference when you look at the ADA and the severe adverse events and anaphylaxis that were reported with one and not the other. And again, for these peptide-related impurities, we're going to be really focusing on how they are presented in terms of MHC, what's the affinity of any impurities for the MHC, and the activation of potential of the TCR. So you know, there's a number of in silico tools that have been developed to do this kind of analysis on primary sequence at least. You know, there are advantages. They're high throughput. They cover multiple MHC types. They can look at potential impurities, not actual impurities that were found in the product. And it can be at least some of the algorithms have information regarding neoepitopes or disruption of tolerogenic sequences. So altogether, it can really give you an idea of whether a new sequence in the peptide could be more or less immunogenic. Weaknesses is that, you know, outside the primary sequence, there's really these methods are a little bit weaker. They are mostly concentrated on HLA-DR. And, you know, again, unnatural amino acids, modifications, side chains, these are things that are not really considered, and of course, no B-cell epitopes. In terms of the in vitro tools, there's a number of them, and we don't have really time to discuss them all, but mostly they're based on the affinity of peptide impurities for the MHC, as well as the assessing the presence of reactive naive T cells, and that's done mostly by DC T cell assays. Again, here the readout can be multiple: proliferation, some cell marker expression, cytokine expression, LE spots. As usual, suitability controls ends up being one of the key aspects where we find a lot of problems. It's very important to remember that we are looking for naive responses, not memory responses. Some studies have used really complex proteins as suitability controls because they are known to activate naive responses. Those have also some limitations because their processing is completely different. There's very limited cross-presentation and so forth. So they're not ideal. What we like to see is a peptide of similar length and general structure containing promiscuous T cell epitopes to confirm the ability of assays to detect naive T cells. And we understand that those probably will not elicit responses from all of the donors. But as long as there's a reasonable amount of reactivity that can be useful. And again, the weakness of this method, of course, the high donor-to-donor variability and the really low frequency of naive T cells. So that's something that really needs to be considered when you're designing these assays. So what to provide the agency, the SOP, supporting data, how the cells are prepared, culture conditions, sample size, and C coverage, on any targeting that you've done to select. Some sponsors will take the in silico studies in select the subjects with the MHC that is of higher risk, and that's an interesting approach. What is the final concentration of the peptides in the well? That's very important. Some sponsors sort of try to use very low concentrations. That's really not advisable. And how you qualified. What are the studies that you've done to qualify the assay so that we can understand the robustness of your assay all around is really important. And of course, what's the readout selection, stability controls? We discussed this already. And what are the qualification exercise that you have done to show that the assay is really fit for purpose? And of course, provide the results. And the results should include for each data point, what was the cell recovery viability data from the suitability controls for the specific assays, and again, numerical results, not positive or negative. And if you can, always we like to see Excel tables so that we can look at data. Common deficiencies that we've seen, I think I've mentioned the number of stream T cells being too low to detect responder naive T cells with any kind of accuracy, inadequate stability controls, and peptide concentration in the wells being too low to elicit a response are three of the main concerns that we've had in previous submissions. So in summary, for generic shows to have the same API as the RLD, track and process-related impurities, they can impact immunogenicity, so it's important that we assess what level of uncertainty they bring to the table. We can use in silico and cell-based methods or results from those assays to help assess the summative effect of different impurities and does help us inform the risk. But they need to rely on assays that have been carefully constructed and demonstrated to be fit for purpose. With that, I want to acknowledge all the discussions and thoughts that went into this talk, particularly Eric Pang, Cameron Smith, Susan Kirshner, and the Immunogenicity Review Committee, as well as people from my lab, Mohan Rekmalangiswaran, Seth Thackard, Jen Herr, Lupin Baker, and the rest of the crew who have been instrumental in developing these assays and the thoughts around them. And I believe we have some challenge questions. So the first challenge question is, an RMI assay SOP should include information on A, cell platform, B, final concentration of DP in the well and any DP manipulations, C, a description of the stability controls included on each run, and D, all of the above. And the answer is, of course, D. For our second question, suitability controls in DCT cell assays should A, distinguish between CD4 and CD8 T cells, B, be very sensitive to innate immune response modulating impurities, C, confirm that the assay detects naive T cell responses, or D, confirm that the assay detects memory T cell responses. And the answer, of course, is C. Very important to make sure we are looking for naive T cell response. Okay, so let me now leave you with one parting thought, and that is that absence of evidence is not the same as evidence of absence, of course, unless the assays are really, really good. So let's strive to have really good assays. Thank you.
Thank you for the great presentations. As a reminder, if you haven't had a chance to submit your questions relevant to the presentations into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Remember to indicate the panelist you are directing the question to by placing the @ symbol in front of their name at the beginning of the question, for example, @Eric. Joining us on the Q&A panel, we welcome one of our session leads, Dr. Cameron Smith. Looks like we have some questions coming in right now, and the first couple of questions will be directed to Dr. Eric Pang. And here is the first question for Dr. Pang. If a generic product has an impurity that is less than 0.5%, but a higher level than the corresponding limit in the RLD, what supporting data is needed to qualify the impurity? And this is a multifactorial question, so we have one more question. What are the studies required in case the peptide impurity is higher than 0.5% and the RLD? And also, has OGD approved any ANDA with peptide impurity levels higher than the RLD?
Thank you. That's a excellent question, and it's one that we're being asked quite often. So first of all, let me clarify that the 0.5% limit is actually for these impurities that are commonly found between the the 0.5% is for the impurities that are new to the generic products. So for these impurities that are commonly found, we don't have this 0.5%. However, the guidance does say that it cannot be more than the RLDs. So for these impurities that are higher than the RLDs, the guidance also says that you need to look at the adaptive, whether that has any MHC binding domains and whether that's going to create additional risk to the product. So with that in mind, you can do similarly what you've done with other impurities that are new. You look at the MHC binding domain of these impurities, whether these are creating a higher risk, and just try to justify that it is higher than the RLD. For the third question, I believe that's something I would defer to Cameron and Yili, since they can speak more about what has been approved so far.
Hi, this is Cameron Smith. So yeah, I can take that last question. So we can't specifically comment on actual approvals, but we can say that for products that are covered by the ANDA peptide guidance that's been referred to in the presentations, that we have reviewed and accepted data for qualification of limits that are higher than what's observed in the RLD for impurities between 0.10 and 0.5%.
Thank you for responding to that question. We do have another question for Dr. Pang, and here is the question. API sameness is required as per the guidance for synthetic peptides. Is sameness also required when the generic API is made from rDNA or a semi-synthetic process?
Thank you for the question. So the question is if API sameness is needed for these other APIs that are made from the recombinant or semi-synthetic process. Typically, they, from a generic product perspective, yes, the API has to be the same. Therefore, sameness demonstration it is required. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We have a couple questions that came in for Dr. Yili Li, and here is the first question. How many lots of RLD are recommended in support of the impurity
Oh, hi. Yeah, this is Yili. So we require at least 3 lots of the ROD with different ages available.
Thank you for responding to that question, Dr. Lee. And we do have another question. And here is the question. How do we capture the age bias in between the test and the RLD if the age difference there is similar while performing similarity?
ROD, the main purpose is to qualify an impurity in the test product. So, I mean, for your benefit, you could use the ROD at the end of the shelf life because that's the worst case. So it may help you qualify your impurity. But if you do the ROD test before expiration date, that will be okay because as you know, if you do the ROD earlier and your test product still like below IOD or similar as IOD, then it will be fine. So I hope I've answered your question.
Thank you for responding to that question. Moving on to our next panelist, we have a couple questions for Dr. Daniela Verthelli. And here's the first question for Dr. Verthelli. For peptides of natural origin, would the immunogenicity risk assessment be more critical for impurities?
Thank you. Good morning to everybody, and thank you for that question. Yeah, we do consider the manufacturing process when assessing the risk of a product. We will look at each impurity individually and try to assess its risk. And products that are naturally derived may have a broader array of impurities, so we will look at them quite carefully.
Thank you for responding to that question. We do have another question for Dr. Verthelli, and here is the question. What is the acceptable percentage variability in the results of cell-based bioassays versus the RLD to conclude the results of the test and RLD are comparable with 15%, within 25%, or as long as they are within RLD range? Is it acceptable?
This is an interesting question. So it really depends on what assay we are looking at and the amount and variability of the data that we have set that we are provided with. I mean, usually you like to see things within 20%, but really what we want to see is that nothing is sort of beyond the range of what we see with the RLD. That's really what we're trying to establish, that the impurities are within that range.
Thank you for responding to those questions. We're moving on to our next panelist, welcoming our session lead, Dr. Cameron Smith. We do have a few questions that came in for Dr. Smith, and here is the first question. For a product with high immunogenicity risk, are innate and adaptive immunogenicity studies required through the shelf life, or is it adequate if a study is conducted by spiking with the maximum specified limit of impurity expected to be present in the product?
Hi, thanks. This is Cameron. Thanks for the question. So yeah, so there's a few things at play with this question. So for a product with higher immunogenicity risk, you certainly need to de-risk or provide risk mitigation strategy for de-risking both the innate and adaptive immune response potential for the product. However, as Yili outlined in her presentation, for the adaptive immune response studies, those studies should be done on the purified impurities of interest. So the shelf life is kind of a moot point for those studies because you're conducting those on the purified impurity. However, for innate immune response, we recommend generally you can conduct those studies on samples that have been aged on a near release and at the end of the shelf life. However, we don't generally recommend that you spike impurities into those samples that you're using to conduct those innate immune response studies. And Daniela perhaps can comment on spiking if she disagrees with what I've said.
Thank you, Cameron. I don't disagree with what you said. The only thing that I would add is that the concentration of the peptide used in the assay needs to be sufficient to ensure that there is antigen presentation. So there is just trying to equate the concentration of the peptide that, well, the peptide impurity that might be in the product may not be a good strategy. We want to really assess the immunogenicity of the impurity. So using it at a concentration that would ensure antigen presentation would be needed. How's that?
Thank you for responding to that question. We do have another question for Dr. Smith, and here is the question. Is the comparative study with the RLD for API sameness and comparison of impurity level relevant only for DP formulation? And also, does any kind of comparison of API with API from RLD have to be done on the DMF level? What kind of characterization is needed for DS on the DMF level?
Okay, thank you again for the question. So again, this is a multi-part question. So generally for the ANDA, we recommend a comparative study for impurity profile be performed on the fully formulated drug product in comparison to the reference product. And so as outlined in the presentations, we generally ask for that on release and at the end of the shelf life. Now for the API, if you're referencing a DMF in your application for the source of your API, generally the DMF reviewer would also ask you to qualify impurity levels that are present or impurity limits that are proposed in the DMF, and that may include a comparison of the API or to a reference product. So obviously that reference product, if reference product A is used to qualify impurities in a DMF, those qualifications would be relevant for an ANDA A that also references that RLD. Regarding the characterization that's needed for the drug substance on a DMF level, so those are generally characterization studies such as that define the structure of the peptide, essentially the primary sequence, any sort of general physicochemical properties such as isoelectric point. There are quite a number of properties that could be characterized. But I probably won't go into all the detail on those in this forum.
Thank you for responding to that question. We do have one more question for Dr. Smith, and here is the question. Is an immunogenicity study required to perform at near release and shelf life? So the question is it needed to perform at an intermediate stability time point?
Yeah, so again, generally we recommend release and shelf life, end of shelf life for these studies. So we would not normally recommend intermediate time points for these studies. Thanks.
Thank you for responding to that question. Moving back up to the beginning of our panel, We have a few more questions that came in for Dr. Pang, and here, Dr. Eric Pang, and here's the first question. For a peptide that is not of rDNA origin, is it possible to qualify specification of greater than 0.5% for an individual impurity where the RLD contains a level of less than 0.5%?
Thank you for the question, and it's similar to the first one that I answered, I believe. So let me clarify. So for these impurities that are commonly shared between the RLD and at greater than 0.5% and it's exceeding the RLD's limit, perhaps it will be justifiable using these in vitro assays. However, it really depends on the totality of the evidence and the nature of the impurities. I think Danella can provide additional insights into this point. Danella?
Hi, Eric. No, I think that you addressed it well. So basically, for new impurities, we prefer not to see anything above 0.5%. Any new impurities. For impurities that are already there, if we see a slight increase, that may be acceptable. Let's say that it's present in the RLD at 0.4%, you have 0.5%. That may be acceptable. You may need some data to look at the potential risk of it, but that would probably be acceptable. It's all about understanding the risk of impurities.
Thank you for responding to that question. We do have another question for Dr. Pang, and here is the question. What about generic synthetic peptide product which refers to the RLD with a synthetic peptide? Do we need immunogenicity studies?
That's also an excellent question. So what about these RLDs with that are synthetic, and then the ANDA is also synthetic? I think it really depends on the risk of the peptides. For some of these peptides that are have a less of immunogenicity concern, the immunogenicity studies are not recommended unless there's a special circumstances for it. Now, that doesn't mean that there aren't ones that do require this. So I think for certain peptides that are higher with a higher immunogenicity risk, perhaps the ideas and the principles from the peptide guidance would be borrowed for these referencing synthetic peptides.
Thank you for responding to that question. Moving on to our next panelist, we have a few more questions that came in for Dr. Yeli Li. And here's the first question for Dr. Li. Do we need to identify impurities that are equal to or greater than 0.1%, even if the level matched with the RLD?
Okay, so the question here actually comes to like how you know the the impurity match with ROD. So we basically need some evidence like the impurity in the test product and ROD are same identity. As I said in the talk, so only through relative retention time is not enough to say like you have the same impurity as ROD. So in that sense, you still need to provide some identity saying the test product and the ROD has the same impurity.
Thank you, Dr. Li, for responding to that question. We do have another question for Dr. Li, and here is the question. Is the identification threshold of 0.5% still acceptable for the peptides not covered by the discussed FDA guideline, and they give in parentheses, which are ANDAs, for certain highly purified synthetic peptide drug products that refer to the listed drugs of rDNA origin. Guidance for industry.
Okay, so the U.S. agency has never accepted the European general guidance for the peptides. So the answer is no, we don't accept EU guidance for. So for any other peptide, it's go case by case, and there's no generally applicable guidance so far.
Thank you, Dr. Lee, for responding to that question. Moving on to our next panelist. We have a few more questions that came in for Dr. Daniela Verthelli. And here is the first question. Does FDA have a list of preferred cytokines, or are sponsors to empirically determine cytokines representative of their product?
So I'm going to assume that we're talking about the innate immune response modulating impurities and how to measure those. There are a number of cytokines that have been commonly used, and there are several papers that have looked at those. Probably one of the latest ones is the one by Holdy et al. So things like IL-1, IL-6, TNF, CXCL10 are sort of the CCL2. Those are regularly used. We really prefer to see multiple parameters assessed. Think about what the potential innate immune paths are and what cytokines are going to be relevant to those. Think about when you are collecting the data and the timing of expression of the different cytokines, and then you can propose a panel that makes sense. It also will depend on the source of the product and how it's manufactured and what the concerns are. So, but in general, cytokines that deal with innate immunity are the ones that we're going to be looking for.
Thank you for responding to that question. We do have another question for Dr. Verthelli. And here is the question. When using a DC T-cell assay, is the right protocol to test all the impurities together, or do they need to be run independently?
Yeah, that's an excellent question. So we really want the individual impurities to be run independently and compared in terms of activity to the RLD, to the API. So yeah, independent assessment of each of the impurities is really what is preferred, and that allows us to put specifications on the individual impurities. We really want to understand which impurities we need to control to a higher degree.
Thank you for responding to that question. Moving on to our next panelist. We do have a couple more questions that came in for Dr. Cameron Smith. And here's the first question for Dr. Smith. If any unspecified impurity is found above 0.1% and cannot be characterized and is also found in the RLD above 0.1%, what studies are suggested, and is it that the impurity considered qualified if supported with RLD levels?
So hi, yeah, this question is a little bit similar to one of the recent questions that Yili answered. So yes, we recognize that there can be situations where it can be very difficult to completely characterize an impurity that may be present at a level greater than 0.10%. So in those sorts of situations, you may be able to get some level of partial characterization. For example, you might be able, so as Yuli said in her answer to this similar question, relative retention time is not enough. So you may be able to get some molecular weight information, maybe some partial sequence information. And if you can make a case based on data such as that, that those two peaks present in the test product and RLD are indeed the same, that may be sufficient. But again, this is a case-by-case situation, and it would be sort of subject to review of the data that was submitted. So I believe that answers the question.
Thank you for responding to that question, Dr. Smith. Moving back to the beginning of our panel, we do have a couple of questions that also came in recently for Dr. Eric Pang. And here is the first question. For peptide products that do not have a PSG yet, how do we know the right risk assessment and the studies needed?
That is an excellent question, and it will take me quite a bit to explain, but I'll keep it short due to the time. Basically for these peptide products that don't have a PSG, I think the first thing one should consider when developing this is looking at the RLD's labeling and find out what are some the known concerns or adverse risk or immunogenicity concerns that they have. So looking at section 6 and 12 of these labeling to find out whether this can be considered a higher risk from an immunogenicity and safety perspective. Then also looking at your product, also how same or similar is it to the RLD also will be very helpful, both from your own testing data and so forth of the impurity levels. Another thing one should consider is, if you still have concerns, please send in, consults for, control correspondence or pre-ANDA meeting requests. I think those are also good ways to verify your thinking. But, we highly recommend that you provide some data as an internal findings and try to justify and say, what do you think this immunogenicity risk is, and whether there's anything that additionally we can recommend. I think those are good starting points. Thank you.
Thank you, Dr. Pang, for responding to that question. We've got just under 2 minutes remaining in our Q&A panel time, and we'll try to squeeze in one more question, and this is for Dr. Pang, and here's the question. For peptides that are not covered as part of the peptide guidance, what are the acceptable impurity limits?
Thanks for the question. Again, this is also can be case by case. I think for these peptides that are depend on the risk of the products, and I'll defer this also to Cameron and Yili to chime in too. But depends on the risk of the product. I think if it's lower risk, perhaps you can using other guidances. I know ICH doesn't apply to peptides. Perhaps the spirit of ICH can. Also, you can use RLD's batch analysis to justify your impurity limits. Cameron and Yili, do you have anything in addition to add to this?
Yeah, I would agree that the first thing you should look at is qualification of the limits by comparative studies with the RLD. And then as Eric described, based upon a risk assessment of the product, you may be able to, if you can't use RLD limits, you may be able to use the spirit of guidances such as ICH, even though they're not officially applicable to peptides. But again, that's going to be case by case. An argument will need to be made, a justification will need to be made in the submission for any sort of argument that you put forward to justify an impurity or setting an impurity limit in a certain way. Thanks.
Well, that's all the time we have for questions. A huge thank you to our panelists for answering numerous questions that came in. We'll now transition to our morning break and return at 10:10 a.m. Eastern Time.